Thank you, Myra. It's a great pleasure to host this webinar today and introduce the speakers. Biosensors have become an indispensable research tool in biology. Having the ability to visualize and quantify biological processes is key for advancing our understanding of how plants function, and of course a foundation for mathematical modeling. Today's webinar will be about exciting new developments on genetically encoded biosensors given to us by two leaders in the field. Xander Jones is a group leader at the Sainsbury Laboratory in Cambridge. He's a leader in plant hormone biology and has published influential papers on hormone signaling and signal integration, and made key developments on high-resolution biosensors. Xander will be joined today by Jim Rowe, who's a research associate in Xander's group with a background in hormones, osmotic stress, and stomatal development. And Jim is currently developing the next generation of biosensors in Xander's group. Both are strong advocates of quantification in plant biology, so it's with great pleasure that I hand over to them to deliver today's webinar. Thank you very much, and we look forward to your presentation.
Thank you very much for inviting us to talk. I'll just share my screen. Share screen. Right, okay. Nearly ready. Cool. So yeah, as Richard said, I'm Jim Rowe and Xander's here as well. And we're going to talk to you today about biosensors. It's going to be loosely based on our paper that we published recently in Quantitative Plant Biology. And what we're going to be talking about is how we can use them to get more quantitative biology, because they're really, really useful tools. We're going to start by discussing the history of fluorescent reporters and biosensors. And really, we're talking mostly about the genetically encoded fluorescent biosensors. There are other things called biosensors and other ways of measuring metabolites or analytes in plants. And they're all really useful, but these are what we're focusing on today. We're going to discuss the benefits of using direct biosensors and how, if they're quantitative, they can be used to sort of analyze biochemistry and signaling in vivo, and the implications this can have for modeling. We then, because we thought this might be a diverse audience and people might be interested in getting started with biosensors, we put a few slides in the talk about tips for getting started with biosensor imaging. And then we're going to talk a bit more about how advanced image analysis can be used to get more info from biosensors. So really, there's two different types of fluorescent reporter or biosensors. There's the intensiometric and the ratiometric. So an intensiometric reporter or biosensor is something where the fluorescence will either get brighter or dimmer dependent on the concentration of your analyte, which is the thing you're analyzing. A ratiometric sensor is something where you look at the two different spectral conditions, and you take a ratio of those two, and that tells you the concentration of your analyte. The difference being that if something is ratiometric, you can get rid of a lot of optical artifacts and things like that. Whereas if you're imaging deep in a plant tissue, for example, things tend to get dimmer. There are various different types of reporters: transcriptional reporters, Degron reporters, direct intrinsic biosensors, and direct extrinsic biosensors. And I think most people in the audience will be familiar to an extent with transcriptional reporter systems, such as DR5. So DR5, what you're assaying when you're looking at your DR5 fluorescence is the activity of the synthetic promoter that's in the construct. In the absence of auxin, what's going on is you've got an AuxIAA repressor that is inhibiting transcription. This AuxIAA repressor is targeted for degradation by the auxin receptor complex in the presence of auxin, allowing transcription to go forward and fluorescence to increase. And these this sort of transcriptional reporter, like DR5, has really been instrumental in developmental biology. We wouldn't know anywhere near what we know now without them. And they really allowed people to sort of see get the first idea of where hormones may be accumulating. And particularly when we started coupling them with fluorescent hormone transporters as well, like the PIN proteins, they allow people to build quantitative and plausible models for hormone distributions in plants, which might explain tropisms or the sites of organ formation, for example. And these models really push developmental biology forward. But if you look at this is quite an indirect measure. Transcription, translation, is quite far away from the site of perception. So in years following, people developed Degron reporters such as D2-VENUS. So D2-VENUS takes a fluorescent venous protein and adds on the Degron domain from one of those AUXIAA repressors. So what this means is that when you add auxin, the protein is quickly tagged with ubiquitin to be degraded by the auxin receptor complex. And what this means, if you look at, for instance, if you look at D2-VENUS fluorescence, is you see a much faster decrease in fluorescence than you see increase in fluorescence with DR5. So you can watch you apply auxin, you see the fluorescence go down quickly within minutes, as opposed to with DR5, where things don't really start accumulating. You can't really see anything for 3, 4, 5 hours afterwards. And this really allowed people to start modeling fast auxin accumulations and things like that, for even more for tropisms and the like. Now, both of the sensors I've talked about so far have been intensiometric. But as you know, if you image deeper, things appear dimmer. And also, like with D2-VENUS, if you've got low fluorescence in a cell, is that due to high levels of auxin, or is it just the protein's not expressed much there? So to get around this, people developed ratiometric versions where you express a non-degradable protein at the same time, in this case, a blue fluorescent protein. And so when you add auxin, you get, you lose your yellow fluorescence, but you maintain your blue fluorescence. So taking the ratio of the two allows you to get a much better grip on how much auxin response there is in a given cell. There is another problem with both of these sensors, though. They rely a lot on the endogenous machinery that's there in the cells, whether that is the auxin receptor, the activity of the proteasome, the transcription, or the translation. And so what we really need is we need ways of measuring hormones, for example, where we can view them, we can view fast responses. The responses are not, they are not affected by the endogenous machinery. Ideally, we'd want it reversible, and often we want it ratiometric as well. And this is really where the direct biosensors come in. There are a couple of different types. There's intrinsic direct biosensors. So Roja GFP is a great example of this. It's an intrinsic biosensor because there's a change in fluorescence due to the fluorescent protein undergoing a change in the due to the thing it's sensing, the analyte. So in this case, it's really sensing the oxidation status. And if you place a Roja AFP in a reduced environment and illuminate it with a blue 488 laser, what you'll see is you get a lot of green fluorescence coming out. However, if you place it in an oxidizing environment, a disulfide forms on the beta barrel, and this causes a lot less fluorescence in that oxidizing environment when you excite with a 488. Now, you can also excite with a 405, which doesn't really change very much. And so taking into account the fluorescence in these two different conditions, you can actually get a ratiometric measure of the oxidation status of the biosensor. There are also extrinsic biosensors because, we can't, ABA doesn't really oxidize GFP. So what we've done here is we've taken a sensory domain. We take a sensory domain, which is separate to the fluorescent proteins. So you have, in this case, you have PIL1, which is the ABA receptor, and ABI, a truncation of ABI1, which is its co-receptor. Now, these two proteins only really interact in the presence of ABA. And so what this means is there's a large conformational change upon ABA binding. And what we've done is we bind them to a fluorescent donor, pair of fluorescent proteins, which we call the fluorescent domains. So we have a big conformational change, but how do we translate this into a difference in measurable difference in fluorescence? Well, we rely on a principle called FRET. So FRET is an energy transfer between compatible fluorophores, in this case, CFP and YFP. And what we find is that this energy transfer is very high when they are close and in the right orientation, but decreases when they move further apart. So it sort of acts as a molecular ruler. And what you can therefore do is if you excite your CFP and there's no ABA, a bit of energy is transferred to the YFP. But when you add ABA, there's a large conformational change. And the same excitation will lead to a lot more energy transfer. So you get a much higher ratio of YFP emission over CFP emission when there's ABA. The nice thing about this sensor is it's sensitive and it is quantitative and it's reversible as well. So, and it's fast. So you can, for instance, pulse a plant root with different concentrations of ABA. And as you go through and then track the emission ratio, which tells you an indicator of the ABA concentration inside the cells. And really you can see not only ABA accumulation, but you can also see depletion afterwards. And what's nice is that if you start looking at this, you can notice that actually after our first treatment of ABA, we start getting accelerated depletion as we go through time. So we think that ABA may be promoting its own degradation or conjugation, for example. Yeah. Now, this is really good because suddenly we can measure the in vivo concentrations of hormones, and we can measure rates of accumulation and elimination. And traditionally, it's very difficult to parameterize experimental model, to parameterize computational models if we want to simulate what's going on in a plant. Whereas now we've got a whole new tool set that we can use to actually start looking at some of these things like the concentrations in individual cells. And these tool sets are really powerful, particularly when combined with things like mutants, inducible expression of enzymes, or hormone precursors, for example. And this is the sort of approach that you used with the GPS-1 sensor recently, isn't it, Xander?
Yeah, so GPS is much like ABACLLY. We engineered it to bind GA. It's a direct extrinsic biosensor. So there's GA receptors that bind GA, and that changes conformation. So you get a FRET change. Just like ABACLLY, it's an increase in ratio. And so one of the first things we did was look at roots and see where's the GA. And yes, we can look at individual cells and model individual cells, but we can also look across tissues. And that's where we need to get to multi-scale modeling. And this is kind of an interesting story because right at the beginning, we saw a pattern of GA accumulation in roots that was actually different than what had been proposed by some mathematical models in 2012. So Leah Band is a mathematician, and she had proposed that actually there would be a certain pattern of GA in the root where there would be high GA in the division zone, the smaller cells, the dividing cells, and that GA would fall to lower levels as it progressed, as you progress to the elongation zone. And that was, interestingly, that most of that model, inspiration for that model was derived from a reporter, a fluorescent reporter that was reporting on the activity of a promoter that drives the GA20 oxidase 1 gene in Arabidopsis fruits. So there was an expression pattern of a biosynthetic enzyme, and that was taken as an indicator of where GA would be. And then the model was proposed to incorporate not just biosynthesis, but dilution, especially as cells get bigger and degradation. So as you were saying, Jim, there's lots of different components that can affect hormone concentrations, biosynthesis, degradation, catabolism, conjugation, and transport, of course, and dilution. So we talked to Leah and we realized that we had the opposite pattern, where actually rather than GA going down in the elongation zone, GA was increasing as the cells progressed to the elongation zone. So already with a direct biosensor, we had a very different view from a sort of indirect reporter. And we needed to know why that was happening. Like, what is actually changing in the cells to give you more GA as you progress, this GA hormone as you progress to the elongation zone. And so we went back and forth with experimentation and modeling with Leah, and Leah created new models, multi-scale cellular models. And in the end, we decided that synthesis was probably a very important driver of the pattern that we were seeing based on certain mutant results and modeling. And then in the end, the key factor in Leah's modeling to predict the GA pattern that we were seeing was actually this factor called alpha. And that's the ratio of synthesis between the division zone and the elongation zone. And the proposal that we're making is that there's a certain relationship, a certain mathematical relationship between this synthesis rates and actually in the end GA levels. And so, yeah, we had there's multiple ways of describing a system mathematically, and Leah found a couple that worked pretty well to describe the system, both before we have a GA, normal conditions, and after you add GA and stuff like that. So once we were pretty convinced that synthesis was an important driver of the system, we wanted to unpack synthesis a little bit more. And so what we can do with GPS is actually combine it with mutants, or as you said, precursor treatments, to really understand in sort of a faster time scale, right? Because we're not looking at, we can look after a few minutes or 20 minutes in this case and see what happens to the GA levels after we perturb biosynthesis. And so the way we decided to perturb biosynthesis is to take the wild type route and add more of different steps of biosynthesis. And that would tell us which ones were rate limiting. So GA12 is actually a precursor that recapitulates the early biosynthetic steps. And we found that actually when we add GA12, we saw more GA, a little bit more GA in the elongation zone, suggesting that one of the early steps is somewhat limiting there. Interestingly, when we added more expression of GA20 oxidase with an inducible expression system, we saw very little change. And that suggests to us that GA20 oxidase is not alone limiting anywhere in the root. And that's really surprising because, as I said, GA20 oxidase was used in Leah's 2012 model as a major reporter because the people thought that GA20 oxidase was the rate limiting step. So what we actually found was that GA3 oxidase, the last step in biosynthesis, was the key step in the elongation zone. So if we overexpress GA3 oxidase, we get a lot more GA in the elongation zone. And so then, that's the sort of information we can really hone in on. It's like, okay, if we're interested in this root pattern of GA in the elongation zone, we should really be thinking about GA3 oxidases. And if we want to understand how it's regulated, we should be really thinking about how these certain GA3 oxidase enzymes are regulated. And that's how the environment can really feed in to regulate a hormone level, is to which enzyme, which step is really the contributing step at any given tissue. And that's an important point because not all tissues are the same. So if you look at the very root tip, when we overexpress GA3 oxidase, we don't see a ton of GA, but when we overexpress both GA3 oxidase and GA20 oxidase, then we see a very high concentration of GA, suggesting that, number one, the early biosynthetic steps are not limiting, but both GA20 oxidase and 3 oxidase are limiting there. And that's the take-home message, is that each tissue is its own entity and different steps can be contributing to the hormone pattern in any given tissue. And when you factor in transport with auxin, it can get very complicated. So that's where the models need to come in to really help us unpick everything.
Yeah. I find this sort of work fascinating, and one thing that also I find interesting is that you've taken you find these rate limiting steps in obviously a single molecular pathway, biosynthetic pathway, but like biosynthetic biosynthesis, metabolism can be very complicated, just like signaling can. And really, we have some advantages sometimes when we look at things like signaling, in that we might have multiple biosensors, so we can pick apart pathways and networks in a way that we can't always with the biochemistry. And in fact, if you look at stomatal signaling, if you look at stomatal closure, under stress, you'll see ROS, you'll see calcium, you'll see ABA induction, and you'll see that a lot of stuff runs through this SNRT kinase, OST1. And actually in the last 20 years or so, there have been numerous sensors developed for ROS, calcium, ABA, and now OST1 activity. And so we're really in a nice point in time where there's a lot of different biosensors out there, and we can start really pulling apart networks in a way that we couldn't before.
A lot of people ask us this a lot, which is like, how many biosensors can you image? Can you image them at the same time? What do you think about that?
So imaging multiple biosensors, it's not always easy, but it sometimes is possible. So one of the main limitations is actually spectral crosstalk. So you can, for instance, image something like a red gecko sensor, which is a cell sensor for calcium, in the same cell as a CFP, YFP fret sensor. But it's more difficult to image two CFP, YFP sensors at the same time. It's just, you just can't really do it.
Yeah, Reiner and Watt had those nice stories with imaging multiple sensors at the same time, when one is intensiometric and one is ratiometric fret sensor. What about imaging two similar sensors but using different compartments to do it in the same cell?
Yeah, you can do that. But then if you're doing that, what you need to do is you need to be really careful with your image analysis so that you can segment those two different compartments more easily. And we'll have a more in-depth discussion about segmentation and image analysis in a little bit. In fact, quite soon, because when me and Xander were talking about doing this talk, we were like, well, I think there'll be a lot of people out there who maybe are starting to image biosensors and and are looking for a bit of advice, or are thinking about starting to image biosensors and thinking about what they can do for them. And really, it's not necessarily always the easiest field to get into. But there are several things that you need to be able to do this kind of quantitative biology. So firstly, you need a good sensor for your analyte of interest. You need a microscope with the right spectral setup. You need a good imaging protocol and a good analysis pipeline. So in terms of a good sensor, there's a whole host of biosensors out there. And Xander, you've spent a lot of time developing biosensors and imaging them. What would you say people should be looking for when they're either developing or imaging, looking for a sensor to use?
Yeah, well, yeah, like you said, we spent 10 years engineering biosensors, and the ABA sensors that we engineered in Abacus, and also at the same time, Reiner developed Avalion. We're the first for hormones, but there's many other sensors in other systems and also in plants. I mean, the first thing people, can obviously think about is the spectral properties. So are you going to use blue and yellow fret? Are you going to use an intensiometric GFP? And there's many, many more flavors. And that's important, but really, your microscope can be retrofitted to whatever output you want. And it doesn't necessarily need to be fluorescence either. There are others. Really, to me, the most important for the end user is really signal to noise ratio. And that's just, whatever dynamic you're imaging, is the dynamic larger than the noise in the system? And some of these are quite noisy approaches, so you need the better signal to noise, the higher that is, the better. Makes it a lot easier to use. And so what we do is, when we're engineering biosensors, is oftentimes the first the first version, the first biosensor isn't so great. We keep making them better. And really that was the nice story with the calcium sensors. They kept getting better and better over time, to the point where now we have really good calcium sensors. And then affinity is super important. You must have the analyte and the sensor has to report on the analyte in the physiological range for whatever condition and cell you're interested in. And that might be different for different tissues or different conditions. So what we often do as engineers is try and engineer an affinity series of sensors so that we can cover multiple different biological phenomenon. And yes, that means different transgenics or different biosensor types. But it's important. And they're also nice controls for each other. So if one is a high affinity sensor, one's a low affinity sensor, they should be responding differently. And that kind of comes to the next point, which is orthogonality. And a lot of people aren't familiar with this word, but we use it a lot in the biosensor field. And it's a really big goal is to have complete orthogonality, or at least get as close as we can. And so the orthogonality means two things. It means the sensor itself doesn't interfere with endogenous signaling or endogenous processes. And, obviously if you're going to bind an analyte directly, like a hormone, you're going to titrate some of the, sequester some of that hormone. So you're necessarily going to interfere with the system at some point. But really that's not the big problem for us in our experience. The bigger problem is overexpressing a receptor protein or signaling protein. So what we have done is engineer orthogonality into various sensors. And other people have done similar things so that the sensor, its expression itself doesn't mess with endogenous phenotypes. But the flip side is just as important. We don't want other signaling processes or endogenous processes to be affecting the fluorescence output of our sensors, such that we're looking at artifacts and not the actual analyte. And sometimes that does happen. And so it's important to have proper controls and be very careful in the interpretation of your data to make sure that you're not looking at something else. And so orthogonality is a really important goal, and sometimes it's imperfect and we kind of keep going until we can get better and better. Something that people don't always factor in when they're starting a new experiment with a new sensor is their microscope properly set up for it? So oftentimes the lasers aren't quite right for the excitations, and you have to have the right filter setups, and there's all kinds of different appropriate imaging approaches with different microscopes. So there's lots of ways to do it. It's not that hard, but you do need to kind of check to make sure you're ready to go when you order your line or new biosensor line seeds. And then as biosensor engineers, we have many experiences with problems in expression. So it's definitely a hiccup that can happen, and we've managed to get around all of them. But yeah, silencing is a big issue that just like anything else. And sometimes you need to try again or try a different line. And sometimes putting a biosensor in a new mutant background, for some reason, your first try is low expression, so yeah, there's all kinds of little, you have to be prepared for some maybe some hiccups along the way when you're getting a new line that isn't already validated for expression. But yeah, like I said, we have solved every sensor we've had in terms of getting it expressed in different mutant lines and in wild type. So that was a journey, but we've made progress.
One I'm sure we'll keep having over and over again.
Yeah.
As we develop more sensors. I think the points that you raise are really important, Xander. And I also think they lead quite nicely into something you touched on, which is the choice of microscope. People always think they need the best confocal to do this stuff on. If you have a very good FP fluorescence, you can do biosensor imaging. So provided you have appropriate filters and a good detector set on there. One of the most important things, though, is the choice of filter sets. So for CFP, YFP imaging, for example, most microscopes don't have excitation about the 430 range and emission in the 535 range that are what you really need for biosensor imaging. It doesn't always come as standard. Often people will pay a bit extra to get it, and you can certainly add it on afterwards, particularly with the EPIs. Sometimes you can get an extra laser line put on your confocal if you have the money. But the best thing to do, actually, when you're considering your biosensor is to actually discuss it with your imaging facility manager, because the chances are somewhat they have a microscope in the department that's kitted out, and or they may well have a microscope that's kitted out for it, or if not, they can give you some advice in how to go about it. And asking for help is always useful, you know. And likewise, once you know that you've got a microscope that can do the imaging, it's useful to start with a good protocol. So this one by Analys in our group is a really nice introduction for CFP, YFP biosensor imaging, and discusses a lot of the problems, the pitfalls, and the ways you can get around them. I would say when you're doing this kind of imaging, there are some things there are lots of things you need to pay attention to, but avoid saturation and dim signals. Always acquire your images at the highest bit rate you can, so 12 or 16 bit, and keep your detector gain even between acquisition channels. And again, ask your facility manager for advice, and they may have some good points for you.
So Jim, this is a lot of stuff, but is you know, that's, for example, ratiometric imaging that different from just imaging a GFP fluorescence?
No, once you've got your imaging set up, it's very, very similar. You're just acquiring an extra couple of channels. And I think it's the initial setup that scares people, but it doesn't take that long to be able to get some quite good data. But I suppose the difficulty then is once you've got your image set, that quite often the thing that takes a lot more time is the image analysis. And I think people underestimate how large a task this can be. To analyze a biosensor image, you might need to do pre-processing steps like blurring to remove noise and background subtraction, separating your channels. You then need to do segmentation. So this is identifying where your biosensor is expressed, and so that you're quantifying just the biosensor, not the background. And you can do this with thresholding and object splitting, or even deep learning approaches. And then you actually need to do for instance, with a ratiometric sensor, you'll need to do your actual ratio calculations. And then you might also want the size and the position of all your different objects. And you can do all of these steps by clicking buttons in FIGI and ImageJ, but it will take you hours and hours to analyze your tool sets, which is why the best thing I can recommend is to go and find easy to use pipelines like this one I developed for FIGI that allow complete segmentation complete segmentation and analysis without much user expertise and input required. So here we can analyze basically every nucleus in a 3D or 4D image, and get the, in this case, ABA concentration in each nucleus. And here you see we basically do it for a whole plant. And then we can label different areas and quantify them individually. So we can do, we can measure stress responses, and we can see that under certain stresses, ABA accumulates higher in the elongation zone than in the root cap, for example. And there are a whole host, this is really nice for nuclear imaging. There are a whole host of tool sets out there for all sorts of different applications. So Joachim Gerthardt has got some wonderful FRET analysis tools for that are really good for cytoplasmic sensors, for example. So if you've got a yellow chameleon calcium sensor, they're really good for that. And there's also comprehensive tool sets to basically analyze every cell in an image, like morphographics, which is a powerful, flexible environment for segmentation, analysis, and reconstruction. And you may think I'm dwelling on this, but the important thing to me is that once you've got a comprehensive image analysis tool set, you can then start combining the biosensor quantification with spatial information, like the size of cells, for example, to correlate cell size against GA concentration, or and use them to inform spatial models. So Xander talked about the model he performed with Leah Band and Annalisa in our group, but there's also some wonderful examples in the literature, things like this is using QD2, which is a Degron-based auxin sensor. And here, DII-VENUS group found a wonderful centrifugal wave of auxin. A really interesting paper. And I suppose the important thing is that this helps close the loop between modeling and experimentation. You know, bring them a lot closer and get the loop much, much faster and closer and more quantitative.
And Jim, like you mentioned this cytosolic and nuclear. What are the consideration sets for, you know, which kind of sensor would you think is useful for what type of application?
Yeah, that's a really good question. So certain signaling events may go on only in certain compartments. So there's some, or the localization of the signaling event may be important. So there's a wonderful paper recently showing that nuclear calcium spike, nuclear calcium is regulating root growth independently of cytosolic. But for the nice thing about localizing a sensor to the nucleus, for example, is that you can segment a cell very, very easily because you're looking at distinct dots. And certain sensors that for certain metabolites that's important, that the localization is important. For others, it's not. So with ABA, we found when we applied ABA, it went up in both the cytosol and the nucleus. So we knew that our nuclear sensors would work.
Mm-hmm.
Yeah, so I'd really like to, well, we really want to thank Annalisa in our lab, who we used lots of her figures in this talk, and our collaborators, Leah and Marcus from Nottingham, and our funders and the rest of our collaborators. Thank you. I'm also going to put a link to the analysis sets in the chat, so anyone who is interested has some protocols that they can download. And I'm going to stop sharing.
Great, thank you very much both. I thought that was a beautifully clear presentation, and fascinating, very educational. Particularly like the way you both played off each other. So many of the questions I actually had, you've kind of asked and answered already in the webinar. So that went really well and learned a lot. I think it was also a really nice demonstration of the power of biosensors for modeling and the importance of software development. So thanks a lot for that. Maybe we just start with a sort of more general question. So, I mean, where do you see the current challenges or limitations for biosensors? Or maybe put differently, what would be top of your sort of most wanted list for biosensors for future development?
What do you think, Xander?
I mean, we have so few biosensors when you think about the big world of all the things you would like to measure. You know, I always get excited about engineering another sensor or other people engineering new sensors. And so, yeah, we have ABA and GA sensors that are published, but we also have other hormone biosensors that are unpublished. And just this year, auxin sensor named Auxin was published, a FRET biosensor for auxin, and we also have a auxin biosensors in development called the Agos. So yeah, and very important to know where auxin is, of course, and more accuracy is better. And there's other types of sensors coming along too. So tension sensors, voltage sensors.
One of the most interesting for me was that the osmolarity sensor, the molecular crowding sensor that
Cesar,
Cesar produced very recently. Doesn't seem to work in Arabidopsis yet, but I'm sure that's coming, you know.
And then, you know, other limiting factors are, you know, just getting enough critical mass where people understand how to use them well. So some people jump in without considering everything. It's actually not that hard once you get a little bit of training, or just, you know, really, but if you're going to follow a protocol, follow it very carefully, I would suggest, so that you can avoid some of the common pitfalls, because it is doing ratiometric imaging in particular is a little more, it's definitely more noisy, and people can run into problems.
Yeah, great. Great, thank you very much. We have a question in the chat, which I'll just read out. So this is from Son Chapaborty. He says, great talk and visuals. Thanks, Jim and Xander. Speaking of microscope, can you use sensors like ABACUS or GPS-1 to look at cross sections of live roots, so without fixing them? So you can look at specialized cell layers, e.g. cortex, which may be sensitive to hormones and required for hormone-dependent cell growth. Great question.
Yeah, I can take this one. We can get a couple layers in, cell layers in with confocal imaging, and a little farther with light sheet imaging or 2-photon, multi-photon imaging. So with the appropriate, if you're driven to, you can get deeper with the current tool sets. But yeah, and the current approaches with actually imaging whole, intact, happy roots. We do think, and we do know that it's possible to cut a tissue and image a cross-section with the caveats that would entail, like the tissue is obviously being perturbed at that point. But I think it's actually pretty effective in a lot of cases. So we don't do a lot of that, but it is definitely possible. And some people really do have to cut their tissues and to look at, for example, axillary buds and stems. We are very much interested in new technologies in the future that can get us deeper or have a different approach. But yeah, I think yes, it's possible. You just have to be aware that maybe the hormone concentrations could be changing after you do the cuts.
Great, thank you. So Jim, you made a good case for ratiometric dyes in the presentation. Are there instances where the intensiometric cases may be preferable, or where there are advantages, or figure out the trade-offs for those?
Yeah, so I think it doesn't all have to, particularly if you're not looking at microscopy as well. Mm-hmm. Some of the calcium, was things like the geckos and stuff like that have very high off and on rates. The signal intensity can increase many thousands of volts, and there's a massive power that comes from that sort of signal to noise ratio. Mm-hmm. And I think if you're looking at a dynamic process like that, like a calcium wave or something like that, then you can, your signal to noise is really high, and you can normalize it to the point before you add the calcium wave. And so, like, sometimes intensiometric approaches can be incredibly powerful. It's
particularly because ratiometric imaging is more noisy, inherently. So if your signal to noise ratio isn't very good, then an intensiometric higher signal to noise reporter can be very useful. So DR5 is actually quite high signal to noise sometimes. And that can be very powerful to give a first look. I think in combination, it's even better to sometimes confirm that it was what you thought it was. And then we always add controls to even our ratiometric sensors. So to make sure that you are looking at the analyte you're looking at, you wanted to look at.
Great. Okay, thanks very much. But I think in the interest of time, we're going to have to stop there. But I really enjoyed that. So thanks for that. Really great talk. Very clear, and again, very educational. Yeah, really enjoyed it. So with that, I think I'll hand back to Myra, and just, thanks again, guys.